Alright, so yesterday we talked about the 3 different sexual life cycles. All sexual life cycles, in other words, any organism that can reproduce sexually, has a time in its life where it's haploid and a time in its life when it's diploid. What distinguishes the life cycles really is kind of how long they spend in the haploid versus the diploid version. So, for example, in animals, the diploid version, after fertilization, our cells undergo mitosis, we become multicellular. Then the haploid version is just sperm and egg. Fungi are the exact opposite of us. Their haploid version undergoes mitosis and becomes a multicellular organism. The diploid version only exists as a single cell. And then plants were the ones in the middle, where the haploid and the diploid version both became multicellular. And you really only need to be able to recognize those from pictures. So today we're actually going to talk about the process of meiosis. Oops. So what meiosis and mitosis have in common with each other, they're both types of cell division. They both happen after S. Remember, in the cell cycle you had, just as a reminder, G1, S, and G2. S was where the chromosomes went from being a stick to being an X. And that has to happen whether we're getting ready for mitosis or meiosis. Meiosis is subdivided into 2 parts, Meiosis I and Meiosis II. It will be important that you know the difference, what happens in each part. And then there's a table at the end of the lab, and this information will help you with that. So one of the differences between them is that in meiosis, the cell divides twice, and you end up with 4 daughter cells. Mitosis only made 2. So mitosis looked like this. You started with 1 cell, you ended with 2 cells. That's mitosis. Meiosis, as you're going to see today, you start with 1 cell, it divides, then these cells divide again, and this is meiosis. You end up with a total of 4 cells. So that is one major difference. Another major difference is that the offspring of meiosis are going to be haploid. They're going to have half the number of chromosomes as the parents. So you're going to go from 2N to N in the process of meiosis. In the process of mitosis, the daughter cells were exact copies of the parent. And even though when I taught you about mitosis, technically I was showing you always 2N or diploid cells. Technically, mitosis can happen in haploid or diploid cells. It doesn't matter. It's just going to make exact copies of of whatever are there. So in mitosis, if the cell started as 2N, it will end as 2N. If it started as haploid, it will end as haploid. So mitosis can happen in diploid or haploid cells. It's just going to make exact copies of whatever's already there. Meiosis only happens in diploid cells for the purpose of cutting their chromosome number in half. It really only happens to make gametes, to make haploid cells that eventually will give rise to gametes. In animals, that would be sperm and egg. All right? So that kind of gives you an overview of the major differences between them. So in the next slide, we're going to start with meiosis I and talk about what happens. And I'll also sketch it again for you just to clarify how it looks different from mitosis. So in meiosis I, this is the step in meiosis where the chromosome number gets cut in half. The chromosomes, rather than lining up in a straight line, they're going to line up with their homologous partner. And rather than the actual chromosomes themselves breaking in half, the partners are going to go in different directions. So the X's are not going to get broken in meiosis I. I know your outline has 2 more blanks on it, and I don't know why, but I deleted what you need for that. So what should go in those blanks is that meiosis I consists of prophase I through telophase I. So just like how mitosis, the names of the phases were prophase, metaphase, anaphase, telophase, PMAT. In meiosis, meiosis I has prophase I, metaphase I, anaphase I, telophase I, and meiosis II is also going to have prophase II, metaphase II, anaphase II, telophase II. And they look kind of similar. So let me show you the difference in meiosis I compared to mitosis. So I'm going to use a cell that has a chromosome number of 4. So this is going to be mitosis on the left. The chromosomes in mitosis broke in half. And the chromatids basically became chromosomes on their own. So now here are our 2 cells at the end. If this cell started as a diploid cell, it ends as a diploid cell. It started with 4 chromosomes. Each new cell ends with 4 chromosomes. And technically these are identical to the cell that they came from, even though these are sticks instead of X's. Because remember, this cell before S, it also had sticks instead of X's. And then when it went through S of the cell cycle, the sticks got a second side. So that's mitosis. Now let me show you meiosis I. So in meiosis I, the chromosomes are not going to line up in a straight line. They are going to line up with their homologous partner. Just like how you made them in the karyotype with their partners, they're going to line up side by side. So in us, that would mean there would be literally 23 pairs lined up in the middle of the cell. And this would be like metaphase. This was metaphase of mitosis. This is what metaphase of meiosis I looks like. Yes? So the purpose of this is that when this divides, the partners are going to separate. So the cells that are made are now haploid. So the whole purpose of this is going to be to make sperm and egg cells, because that way the sperm and egg will have half the chromosome number, and then it'll get restored when fertilization happens. So this is only happening, meiosis, to make basically gametes. In any body cell, you're never ever going to see meiosis happen. All right, so now we went from 2N, 4 chromosomes, to N, and in this case, that's 2 chromosomes. In us, that would be taking it from 46 to 23. Now remember I told you that the total number of cells made is going to be 4. This is only meiosis I. There's a second step, and these are going to split again. So we'll talk about that in a second. Yes? No. As a matter of fact, that's actually what I'm going to show you. This may answer your question. If I don't answer it, ask me again after I show you this. So this is really important, this little statement here. They sort independently. In the next chapter, we're going to learn about Mendel's laws. One of Mendel's laws is the law of independent assortment. So I want to explain what happens in independent assortment. So I'm going to switch over to this picture I already drew. So let's say that this is some organism, and this is the adult. The adult organism is diploid, and its gene code is big B, big S, little s. B is a particular gene that's carried on this chromosome pair, and S is carried on this one. And if we can assume that black is dominant and spotted is dominant, this one is black and spotted. So this is what independent assortment means. So when meiosis happens, these chromosomes are going to line up side by side. They carry information about the same thing, fur color, but they don't carry the exact same information. So when meiosis happens, it's going to give 1 copy of B and 1 copy of S. Let's just say this is a male. This is going to make sperm. So it could give the big B and the big S, which would mean this sperm would be carrying the genes for black and spotted. It would not be carrying white or non-spotted. Now, depending on what egg it fertilizes, that's how you'll get what the offspring will look like. What's another possible combination besides big B and big S? Little b and little s. It could give this B with the little s, and so this sperm would be carrying white and non-spotted. There's still 2 more possibilities. What else? Good. Big B with little s, and what's the last one? Little b, we got big this. So from this one parent, there's actually 4 possibilities of what they might give to an offspring. This is what independent assortment means. When we say they sort independent, it means it's completely random which copy of chromosome 1, which copy of chromosome 2. Big and big is here. Little, big, little big. Yeah. So, here's my point. There's a mathematical way to calculate how many possible outcomes there are based on the haploid number of the cell. So the haploid number is 2, right? There's 2 different chromosomes. It's 2 to the n, with n being the haploid number of chromosomes. So in this case, 2 to the n, that means there were 4 possibilities. In humans, we have 23 chromosomes, a haploid number. So 2 to the 23rd. That means the chance of 2 sperm, just from one guy, being the same, is 1 over 2 to the 23rd power. Almost impossible. That's how many different possible combinations of chromosome 1, chromosome 2, chromosome 3 there are. That's why it's probably unlikely that you have an identical gene combination to any other person on the entire planet, even though they say you probably have a twin somewhere. You don't have a genetic twin somewhere. I mean, unless you're an identical twin. Because it's 1 and 2 to the 23rd of having 2 sperm be completely identical from the same person. And it's the same 1 and 2 to the 23rd chance of having 2 eggs be the same. So the chance of a person being identical to some other unrelated person is almost completely impossible. Yes? Yeah, I mean, even with, but not identical twins. The reason why identical twins are the same is because the split that makes them twins happens after the sperm fertilizes the egg. So it's still 1 sperm and 1 egg. They get fertilized, it undergoes mitosis, and those somehow get broken apart, and basically each one becomes a baby on its own. I mean, in theory, if you had a zygote and it underwent, let's say, 2 divisions, and then somehow something happened that those 4 cells that were made got separated from each other, you would have quadruplets that would be identical, clones of each other. I mean, that's basically what's happening with identical twins.
So then what's happening with the twins that like born at the same time?
Yes, so fraternal twins. So what's happening there is, they're no more closely related than you and your sister. They just happen to get fertilized at the same time. So obviously males release 1,000, or no, more than that, it's like 1 million sperm. If the woman releases multiple eggs that month, and they both are 2 or 3, and they all get fertilized, that's how you'll get fraternal twins. So they are no more closely related than having babies at different times. They just happen to have both of them at the same time. So that's the difference. All right, so that's what independent assortment means. Hopefully that that clarifies that statement. And I do have the terminology here. Here. So 2 to the n is the number of possible combinations that you can have from independent assortment. So whatever the haploid number is, 2 to that number is all the possible sperm and eggs that can be made. And that's why you're not going to see, 2 that are exactly the same. It might be a little more likely in, something like a fruit fly that only has 8 chromosomes. But even then, they all look alike to us. But even then, they're probably not going to be exactly the same. So at the end of meiosis I, we've only divided once. And we've made 2 cells with the haploid chromosome number. 2 cells that have half the original number of chromosomes. So that's meiosis I. Meiosis II is going to look more like mitosis. Anybody still need this? So a couple more writing. So I'll pause real quick and let you guys finish. Okay, so meiosis II looks a lot like mitosis. The chromosomes are going to split at the centromere. In other words, they're going to break in half like they did in mitosis. So it's going to look like this. This was meiosis I. This was our pairs. I'll just put an M1 here. Meiosis I. The chromosome number got cut in half. In meiosis II, the actual chromosomes themselves split at the centromere. Nothing has technically changed as far as the number of chromosomes. This went from 2n to n, but these are still n. These are still haploid. We haven't cut the number of chromosomes in meiosis II. Now, so at the end of the whole thing, you should make 4 cells. But that doesn't make a lot of sense, because if that was true, shouldn't females have babies in 4s? I mean, in theory, if every month you release an egg and it goes through meiosis and forms the mature eggs, shouldn't you have 4 babies at a time? That's what this is basically saying. So it turns out that when eggs are made, it's a little different. So in spermatogenesis, 4 sperm are made by meiosis, just like I drew. But in oogenesis, which is the making of eggs, what happens is the cytoplasm doesn't divide evenly. So meiosis I happens, all the cytoplasm goes to 1 egg. And then when meiosis II happens, all the cytoplasm goes to 1 egg again. These other 3 disintegrate. These are called polar bodies, and they basically just disintegrate and get reabsorbed. That is why if you've ever seen a video of like a sperm fertilizing an egg, the sperm is like this little tiny thing and the egg is huge. All the cytoplasm of you and the mitochondria and all the cell parts come from the egg, not from the sperm. So that's why oogenesis is different than spermatogenesis. 1 egg per every 4.